Welcome to This Explainer. Today, we're stepping into the totally fascinating and, well, sometimes a little tragic world of botanical forensics. We're going to uncover the true culprits behind plant damage, focusing specifically on a highly misunderstood area that catches so many of us off guard, and that's diagnosing chemical injury in our plants.
We've literally all been there, right? You walk outside, or maybe just into your living room, only to discover a withered, dying plant. And our immediate instinct is usually to blame a devious bug or some rampant spreading fungus. But before you do that, we need to pause, put on our detective hats, and examine the clues. We can't just guess here. We really have to let the evidence speak.
First up in our investigation, the suspect lineup. Biotic versus abiotic.
So this basically splits the world of plant problems right down the middle. On one side, your biotic suspects are the living organisms. We're talking fungi, bacteria, viruses, and insects that actively attack specific plants. On the flip side, we have abiotic culprits. These are non-living environmental stressors. This includes things like extreme weather, mechanical damage, like, say, accidentally whacking a tree trunk with your weed whacker, and crucially for our investigation today, chemical exposure.
To accurately identify our killer, professionals rely on the 6-step investigative framework.
Instead of blindly guessing, we have to follow the evidence systematically. This framework starts with identifying the exact host plant, then mapping out the visual problem and its distribution, tracking the timeline, and finally evaluating the context. Things like checking for recent chemical applications nearby. It's a really structured way to rule out those usual biological suspects and see if something more synthetic is actually at play.
Next up, mapping space and time.
These are absolutely critical clues, and this breakdown really highlights the dividing line between living and non-living problems. Think about it. If a living pathogen is the culprit, the damage is usually scattered and progresses slowly over time. But if the damage is uniform, if it follows straight mechanical lines like an irrigation row, appears super suddenly, and hits totally different plant species all at once, well, that points squarely to a non-living event.
So the key takeaway here is that abiotic events leave a very distinct, undeniable signature. They hit fast, the damage is completely uniform, maybe affecting just the upper foliage or a specific side that was facing the wind, and they do not spread to new, healthy growth once the initial event is over. And actually, very often these sudden, uniform, non-living injuries aren't acts of nature at all. They're human-caused chemical accidents.
Which brings us to the real plot twist of our botanical whodunit: chemical sabotage.
If you look at this pear tree, notice how the dead, brown leaves are clustered together. This sudden, uniform browning is not some relentless spreading disease. This is phytotoxicity. Specifically, it was caused by dicamba herbicide drifting over on the wind from a nearby application. It happened fast, and it scorched the exact side of the tree that was exposed to the drift.
Now, tomatoes happen to be incredibly sensitive barometers for herbicides in the environment, particularly chemicals like 2,4-D and dicamba. When they're exposed to off-target drift, or even if they're planted in compost contaminated with pasture herbicides, they don't necessarily just turn brown right away. Instead, they exhibit severe leaf curling, cupping, and a very distinct, weird downward bending of the leaf stalks.
This twisted symptom actually has a specific name: epinasty. It's an abnormal, downward curving growth, and it happens because the cells on the upper side of the leaf stalk suddenly grow way faster than the cells on the lower side. This is a massive red flag. When you see this specific twisted distortion, it alerts you that the plant has almost certainly suffered from chemical exposure rather than your typical garden pest or disease.
Let's contrast this, just to make sure we don't misdiagnose things out in the field. While a fungal disease slowly forms expanding target rings, dry lesions with clear borders, and eventually produces visible signs like fuzzy spores, a chemical burn is an entirely different beast. A chemical burn doesn't grow rings, and it definitely doesn't leave fuzzy spores. It just scorches, and then it stops.
But herbicides aren't the only chemicals causing chaos. Let's look at the salt burn signature.
Excess fertilizer salt is a super common non-living weapon, and it leaves its own highly specific fingerprint. If you want to know if you've over-fertilized, look for marginal browning, also known as necrosis, that begins right at the leaf tip and proceeds down to the base along the very edge of the leaf. And here's the real giveaway. Notice how the dead tissue is separated from the healthy green by a slender yellow halo. If you see that pattern marching uniformly along the leaf margins, you've officially found the calling card of salt burn.
Moving on to indoor investigations: household chemical sensitivities.
Because hey, These silent crimes happen right inside our living rooms, too. Indoor plants can be incredibly sensitive to chemicals we don't even think twice about. Fluoride and chlorine in everyday tap water are major stressors. Even household cleaning sprays or leaf polishing products can cause severe reactions. Literally, trying to make your plant look shiny might actually be poisoning it. For highly susceptible plants like dracaenas, maranthas, and spider plants, their tips will quickly burn and turn brown before the rest of the leaf even begins to curl if they're exposed to these common household chemicals.
So, how do we protect our sensitive suspects? It basically just comes down to simple environmental management. Let your tap water sit out in a pitcher for 24 hours to allow chemicals like chlorine to off-gas before you water. Or, better yet, just switch to filtered or rainwater. Keep cleaning aerosols far away from the foliage. And if you need to dust your plants, just wipe them with a soft cloth and plain water. And if you suspect an accidental chemical spill actually reached the pod, flush the soil thoroughly with clean water.
As you build your case, whether you're working indoors or outdoors, always remember this golden rule of botanical diagnostics from the CSU Extension. I absolutely love this quote. It reminds us, don't jump to conclusions, because multiple, completely different problems can cause identical wilting or yellowing. Let the symptoms and the spatial and temporal clues lead you to the diagnosis. Never try to force a diagnosis to fit just because you found a single random bug on a leaf.
So, the next time you instinctively want to blame a hungry caterpillar or a stealthy fungal spore for your dying plant, step back. Look at the environmental clues. Check the timeline. Check the distribution. Check for twisted epinasty or those telltale saltburn halos. And ask yourself this somewhat uncomfortable question. When you really look at the evidence, could you actually be the prime suspect?